Alright, so we are done with pharmacogenomics and also personalized medicine. The last topic would be stem cells. Yeah. Stem cells can morph into all manner of specialized tissue cells, and these cells have potential to treat many kinds of medical conditions, as well as serving as a testing ground for drugs. Stem cells are distinguished from other cell types by two important characteristics. First, they are unspecialized cells capable of renewing themselves through cell division, sometimes after long periods of inactivity. And second, under certain physiologic or experimental conditions, they can be induced to become tissue or organ-specific cells with special functions. There are three main types of stem cell: embryonic stem cells, adult stem cells, and induced pluripotent stem cells. Embryonic stem cells supply new cells for an embryo as it grows and develops into a baby. These stem cells are said to be pluripotent, which means they can change into any cell in the body. Adult stem cells supply new cells as an organism grows and to replace cells that get damaged. And they are said to be multipotent, which means they can only change into specific cells in the body, not just any cell. For example, blood or hematopoietic stem cells can only replace the various types of cells in the blood. And skin or epithelial stem cells can provide the different types of cells that make up our hair and skin. Induced pluripotent stem cells are stem cells that scientists make in the laboratory. Induced means that they are made in the lab by taking normal adult cells like skin or blood cells and then reprogram them to become stem cells. Just like embryonic stem cells, they are pluripotent, so they can develop into any cell type. There are many potential stem cell applications. Okay, for example, research tools. The use of stem cells as research tools within a laboratory setting will be useful application. The use of stem cells as cellular therapeutics within the human body, mediated via cellular regeneration, paracrine signaling, or modulation of the environment. Source materials for 3D printing and 3D weaving, whereby the stem cells within 3D printing applications, including 3D printing of tissue or organs that are seeded with living cells, or integration of stem cells into 3D printing inks. For drug target validation, this will allow the validation of the predicted target using tissue-specific stem cell-derived cells. And for drug delivery, the delivery of therapeutic products via stem cells and also stem cell exosomes is one of the applications. For the toxicology screening, stem cells can evaluate the effects of drugs on our biological systems. So, stem cells are very useful in regenerative medicine. The 6 classes of stem cells, that is, embryonic stem cells, tissue-specific progenital stem cells, or PSC, mesenchymal stem cells, MSC, umbilical cord stem cells, UCSC, bone marrow stem cell, BMSC, and induced pluripotent stem cell, iPSC. They all have promises in regenerative medicine and also disease therapeutics, as depicted in this figure.